Hey LeBron, could you tell me a little bit about the activation of the NF kappa B complex?
All right, so let's walk through how the NF kappa B pathway is activated. In resting cells, NF kappa B, which is a transcription factor, is held inactive in the cytoplasm. It's bound to an inhibitor called I kappa B alpha. This inhibitor masks the nuclear localization signal on NF kappa B, which prevents it from entering the nucleus and doing its job.
Right. I get the basic concepts, but can you break that down a little bit more for me?
So when a cell detects a stimulus, this could be something like tumor necrosis factor alpha, interleukin 1, or pathogen-associated signals through toll-like receptors. That signal is received by membrane-bound receptors. These receptors then initiate intracellular signaling cascades.
LeBron, you mentioned the tumor necrosis factor alpha. Can you give us a little bit more insight on that as the specific stimulus?
So TNF binds to its receptor, which recruits a set of adaptive proteins, including TRAD and TRAF2, to the intracellular domain. These adapters help transmit the signal downstream by activating other intermediates like RPK1 and TAK1. TAK1 then activates the IKK complex. This is a key step. The IKK complex is made up of three proteins, IKK alpha, IKK beta, and a regulatory subunit called NEMI or IKK gamma. The active IKK complex phosphorylates I kappa B alpha on specific serine residues. That phosphorylation acts as a signal for ubiquitination, which tags I kappa B alpha for degradation by the 26S proteasome. Once I kappa B is degraded, NF kappa B is released. At this point, the free NF kappa B, typically a dimer of P65 and P50 subunits, translocates into the nucleus. There, it binds to specific DNA sequences and promotes the transcription of target genes involved in inflammation, immune response, cell survival, and proliferation. Importantly, one of the genes it turns on is I kappa B alpha itself, which gets translated and comes back to the cytoplasm to bind NF kappa B again, essentially resetting the system. That's part of a negative feedback loop that helps regulate the duration of NF kappa B activity.
So, LeBron, is that the only way that NF kappa B can actually be activated?
There's also a non-canonical pathway, which is slower and uses different components, mainly NICI and IKK alpha, to process P100 into P52 and form P52/RelB dimers. This branch is typically involved in developmental processes and long-term immune regulation. So in summary, the activation of NF kappa B is a tightly regulated process that allows the cell to respond quickly to stress or infection, but also turn that response off once the threat is resolved.